Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over integration of metabolic pathways, especially metabolism that is going on during fasting and starvation. Okay, now let's get into the details. So before we understand the details on the metabolism that is going on during fasting and starvation, let's try to understand some of the terminologies that we use. So we have something called as postprandial state, and that is 4 hours after the meals, from the point of taking meals, from that point until 4 hours from there, it is a postprandial state. And then the post-absorptive state. Post-absorptive state is 6 hours from the postprandial state. It is a post-absorptive state. And the fasting state, it is 10 hours from the point of meal that is taken, that is 4 hours. Postprandial, 6 hours post-absorptive, and from there onwards, so that is the fasting state. So fasting state, generally it starts 10 hours after the last meal. So these are some of the terminologies that we use, and also you must be aware of the terminologies that we use. And also note that, so when we call a breakfast, so that means we already there in the fasting state by that time, and you're going to break that fasting by taking breakfast. That's why the terminology breakfast is used, because we truly break the fast, because by that time it would be more than 10 hours after the last meal. Okay, so absorptive state is 4 hours, up to 4 hours after meals. Post-absorptive state is next 6 hours from the end of the absorptive state. Fasting state is from 10 hours after the meals until 24 hours. That is what is a fasting state. And the prolonged fasting state, it extends from 24 hours until 72 hours. That is 3 days. And then 3 days onwards, like 72 hours and after, we call it as a starvation state. So these are some of the timelines that you must be aware of. Now, what are the metabolic changes that goes on during post-absorptive fasting and starvation? So, whenever we go into post-absorptive fasting and starvation, it's all about glucagon and lack of insulin. So, very first, the process that starts when we go into post-absorptive state, it is the glycogen breakdown, glycogenolysis, which will start 2 to 3 hours after meals, peaks by 12 hours, and total glycogen will be depleted by 24 hours. And then once this happens, by 12 hours of going into post-absorptive state and fasting, so gluconeogenesis steams up, it becomes peak by 24 hours, and from there onwards it will be staying there for at peak for another 4 to 5 days, and slowly it drops to a certain level, and then it is maintained at that particular level. So gluconeogenesis, it will be at peak in the first week of fasting and starvation, and then it dips in the second week and maintained in that particular phase. So as this glycogen degradation and gluconeogenesis is going on, so blood glucose levels under fasting conditions, so basically it drops, it drops there, and then it maintained there, because gluconeogenesis is maintained at a constant level from second week onwards, and that's how the blood glucose level also kind of falls, and then it is maintained around lower range of fasting level, which is around 70 to 80 milligram per deciliter. Now, as this process is going on, so the fatty acid slowly rises in the blood. So as we move into post-absorptive and fasting state going into starvation, as the fatty acids are rising in the blood, so ketone body levels will also rise, especially 24 hours after and going into 3 days, that is 72 hours. A significant number of ketone bodies can be seen in the blood, and then they continue to rise for another 2 to 3 weeks, and from there they stay at a constant level as long as person is fasting and surviving, and starvation and surviving. So these are some of the changes that are going on there in the blood levels. Now let's see what are the metabolic changes that happens during post-absorptive state and fasting state. Okay, that means until 4 hours and then until 10 hours. Basically, from the point that we take meal until 10 hours of the last meal, what are the changes that goes on? So, obviously, we are in post-absorptive and fasting state, means glucose level will be dropped, insulin level decreases, and glucagon level increases. So when this happens, so glucagon is the predominant hormone that is working here. So what glucagon does? Glucagon is going to break glycogen in the liver, and that glycogen is released as glucose, and glucose is released into the blood, and that glucose in the blood is taken up by the brain, and that it is used for energy purpose, in that where it is converted to acetyl-CoA, T-cell cycle and energy. And also red blood cell use that blood glucose and for its own energy needs. So it is the brain and the red blood cells which are predominantly using the glucose coming from glycogen breakdown. And red blood cell releasing glucose as pyruvate, pyruvate as lactate, and lactate comes out of the red blood cell and it is taken up by the liver, and it will be lactate is converted back into glucose. So when this is going on, even the adipose tissue which is undergoing lipolysis, that is triacylglycerol present in the adipose tissue, it is broken down into glycerol and free fatty acids. Glycerol reaching liver is undergoing gluconeogenesis process to make glucose. Similarly, proteins present in the skeletal muscle which will be released as amino acids, and predominant amino acid that is coming out of the skeletal muscle is alanine, and the next predominant one is glutamine. So the alanine, glutamine, so they are coming into the liver where their nitrogen is taken out, and they will be making urea from the amino group of amino acids, and urea is released into the blood, and that will be excreted out of our body in the urine. Whereas the carbon skeleton of alanine and glutamine will be converted into glucose, and glucose is released into the blood. So overall here, gluconeogenesis is supporting the blood glucose needs of the brain and the red blood cell. Okay, so the predominantly the glucose is coming from glucogenic amino acids that are getting into the liver and being converted into glucose, while their amino group is converted to urea. So what about the fatty acids coming from adipose tissue? So fatty acids which are flooding into the blood, so taken up by the skeletal muscle for their use, cardiac tissue, they will use fatty acid upon oxidation or beta oxidation of fatty acid. Some of the fatty acids that are taken up by the liver initially given for energy purpose and converted to acetyl-CoA, and so because the fatty acid flooding into the blood is not that much, it is not a significant one. That's why generally ketone body formation at this stage, that is post-absorptive and fasting state, it is relatively less here so far. Okay, so as this fasting continue, then there will be rise in the ketone body. We are going to see that later. Okay, just to summarize post-absorptive and fasting state metabolic changes. So the glycogen degradation peaks at 12 hours and depletes by 24 hours, and it will provide glucose needs of red blood cells, brain and other neuronal tissue. Gluconeogenesis peaks by 24 hours, while the glycogen degradation is totally depleted by 24 hours. So it will be at peak, gluconeogenesis is at peak for another week, and then it will drop and maintain a certain level from there onwards. So gluconeogenesis, it provides glucose needs of red blood cell, brain and neuronal tissue. Now what are the substrate for gluconeogenesis during post-absorptive and fasting state? It is the alanine that is coming from skeletal muscle degradation, and alanine coming from the skeletal muscle as such. Along with alanine, glycerol will come from adipose tissue triacylglycerol breakdown. Lactate will come from anaerobic glycolysis. These are the gluconeogenic substrates that can provide glucose needs during post-absorptive and fasting state. To a certain extent, it can be propionyl-CoA coming from oxidation of odd number carbon fatty acids also. Now, fatty acids from adipose tissue rises slowly in the blood and undergoes oxidation. Muscles start using fatty acid for energy. Liver start diverting acetyl-CoA into ketone bodies, although at this time significant appearance of ketone bodies in the blood may not be seen. Now let's see what are the changes that are going on during prolonged fasting state. Prolonged fasting state, remember, it is between 24 hours of hour from the last meal until 72 hours. That's what is prolonged fasting. So glucose is down, insulin is down and glucagon is up. So again, by this time glycogen degradation is totally done. So there is no glycogen that is left which is contributing to glucose, no role of glycogen there. and glucose is coming from gluconeogenesis, predominantly skeletal muscle degradation releasing alanine. Alanine is carbon skeleton of alanine providing glucose, and the amino group is converted to urea. So gluconeogenesis is going on. As and more gluconeogenesis is going on, more and more urea is formed, and urea is excreted in the urine, while glucose is released into the circulation by the liver, and that is predominantly taken up by the red blood cell. And still brain is using glucose for its energy need, although it is trying to adapt itself to use ketone bodies at this point in time. So brain uses both glucose and ketone bodies at this time. Red blood cell uses only glucose, releasing lactate, and lactate is taken up by the liver and converting that back into glucose. Now, what about adipose tissue? Adipose tissue, triacylglycerol breaking down into glycerol and fatty acid, and glycerol is converted to glucose, while fatty acids are flooding into the blood, taken up by the skeletal muscle. Skeletal muscle continue to use fatty acid for their energy purpose, although they are also using ketone bodies along with the fatty acids. So, and the fatty acids taken up by the liver, conducting beta oxidation and diverting most of those acetyl-CoAs in the liver into ketone bodies, and those ketone bodies significantly appearing in the blood now, and those ketone bodies are now starting to take up by the brain. So brain start to use ketone bodies now in the prolonged fasting condition. It is starting to adapt itself to use ketone bodies, while skeletal muscle also significantly using ketone bodies like other tissues. Okay, so here in prolonged fasting condition, so the brain and extrahepatic tissues start to use ketone bodies along with the fatty acid. Of course, brain do not use fatty acid, it uses glucose and ketone bodies at this point in time, whereas skeletal muscle uses both ketone bodies and fatty acid. Similarly, cardiac tissue can also use fatty acids and ketone bodies in prolonged fasting condition. Okay, so just to summarize what we have seen in this particular figure. So prolonged fasting, it extends 24 hours after meals until 72 hours from the last meal. Glycogen is totally depleted by now and do not provide glucose into the blood. Gluconeogenesis is at peak. It provides glucose for RBC, brain and other neuronal tissue. Substrate for gluconeogenesis is mainly amino acid, alanine coming from skeletal muscle. Skeletal muscle degradation has increased. Glycerol from adipose tissue, lactate from anaerobic glycolysis, propionyl-CoA if there is any. So they all will contribute to gluconeogenesis. Now fatty acids from adipose tissues rising in the blood undergo oxidation, ketone bodies start appearing in the blood significantly, and these ketone bodies are used by the muscle along with the fatty acids. Brain start to use ketone bodies along with the glucose. Now let's see what is the metabolic adaptation that is going on during starvation. So glucose continue to be low, insulin is decreased and glucagon is increased. So glycogen is totally depleted by this time. So gluconeogenesis, look at gluconeogenesis. Gluconeogenesis is also kind of decreased after 1 week of starvation. So that means the gluconeogenesis is supported now by glycerol coming from adipose tissue lipolysis, lactate coming from anaerobic glycolysis, and also some of the propionyl-CoA coming from oxidation of fatty acids. So these are the things that can support gluconeogenesis, and glucose that is coming out of gluconeogenesis, which is relatively less now, so that will be taken up purely by the red blood cell, because brain has adapted to use ketone bodies at this time. That's why brain is not taking much of glucose now. It is sparing that glucose for red blood cell. Now as the fatty acids are flooding into the blood, so fatty acids taken up by the liver is going to conduct beta oxidation, releasing acetyl-CoA making ketone bodies, allowing ketone bodies significantly getting into the blood, and most of those ketone bodies are now taken up by the brain, because skeletal muscle decreases its use of ketone bodies by increasing use of fatty acids for their energy purpose. So skeletal muscle is going to spare ketone bodies for the brain, because brain is sparing glucose for the red blood cell. Okay, this kind of metabolic adaptation is going on during starvation, so that the energy resources are used optimally. Okay, so here is the summary of what goes on in the starvation state. So this starts from 3rd day onwards and continue until food is consumed or until the death happens. Okay. So gluconeogenesis drops by the end of a week as muscle protein breakdown decreases and continue to provide glucose for RBC's brain and neuronal tissue. So substrate for gluconeogenesis right now is glycerol from adipose tissue, lactate from anaerobic glycolysis. So fatty acids from adipose tissue continue to rise in the blood, undergo oxidation, ketone bodies continue to rise in the blood significantly. Muscle will use both fatty acids and ketone bodies until 2 weeks of starvation, and then muscle will decrease its use of ketone bodies and spare them for the brain. While brain completely adapts itself to use ketone bodies, so it uses ketone body for its energy purpose and it will spare glucose for the red blood cell. So this kind of metabolic adaptation between the organs will go on, and the energy is kind of shared among all the tissues for the survival. Okay, so now let's look at what is the role of epinephrine and cortisol in the integration of metabolic pathways. Let's look at epinephrine first. So epinephrine increases release of glucagon from the alpha cells of pancreas, and it will decrease the release of insulin from the beta cells of pancreas. So overall, presence of epinephrine during starvation and fasting so lead to elevated glucagon level. It increases degradation of glycogen in the liver and skeletal muscle. It increases lipolysis by degrading triacylglycerol from the adipose tissue, and also it increases gluconeogenesis. Whereas cortisol, what it does, it increases protein degradation in the skeletal muscle, decreases the protein synthesis, and it decreases the utilization of glucose in the muscle and adipose tissue, thereby try to bring up glucose level and spare it for other tissues that needs glucose. And also cortisol increases lipolysis by degrading triacylglycerol from the adipose tissue. It increases gluconeogenesis, and most importantly, cortisol also increases glycogen synthesis by keeping glycogen synthase active. Now, what are the enzymes, important enzymes that are active and inactive during fasting state and starvation? Now, the active enzymes during fasting state and starvation are glycogen degradation enzyme, that is glycogen phosphorylase, glucose 6-phosphatase enzyme, which is a gluconeogenic enzyme, fructose 6-bisphosphatase, which is a gluconeogenic enzyme, phosphoenol pyruvate carboxykinase, pyruvate carboxylase, all of them are gluconeogenic enzymes. Glycerol 3-phosphate dehydrogenase, glycerol kinase, these are all converting glycerol into dihydroxyacetone phosphate, making gluconeogenesis possible. And adipose tissue lipolysis enzyme, hormone sensitive lipase enzyme, is active. Carnitine palmitoyl transferase 1 urea cycle enzyme is active under fasting state. Mitochondrial HMG-CoA synthase enzyme, which is a ketone body synthesis enzyme, which is active. Carbamyl phosphate synthetase 1 enzyme, which is in the carnitine palmitoyl transferase 1 enzyme, is a enzyme involved in fatty acid oxidation, transport of fatty acid, long chain fatty acid into the matrix of mitochondria. And NAC synthase, N-acetyl glutamate synthase enzyme, is also active under fasting state and starvation. Now, what are the enzymes that are inactive under fasting state and starvation? So the inactive enzyme, So inactive enzymes are glycogen synthase, glucokinase hexokinase, PFK1 and PFK2 enzyme, pyruvate kinase, pyruvate dehydrogenase complex, citrate lyase, malic enzyme, acetyl-CoA carboxylase, fatty acid synthase, HMG-CoA reductase, glucose 6-phosphate dehydrogenase, glycerol 3-phosphate acyl transferase, and lipoprotein lipase. All these enzymes are basically synthetic enzymes. Directly or indirectly, they participate in synthesis process, reductive biosynthesis process, which are induced by insulin. So all of them are inactive now. So any enzyme that is activated by glucagon, epinephrine, cortisol, they are all active. Any enzymes that are activated by insulin, right now they will be inactive. Remember that point. So that is all about integration of metabolic pathways during post-absorptive fasting and starvation. So the metabolic changes here, glycogenolysis, which is peaking at 12 hours and depletes by 24 hours. Gluconeogenesis, which peaks by 24 hours, continue at almost at peak by for another 5 to 7 days, and then it drops to continue at some basal level. And blood glucose initially maintained by glycogen breakdown, and then it is maintained by gluconeogenesis. Ketone bodies coming from fatty acid oxidation, it is supporting ketogenesis, and ketone body appears after 24 hours, and significantly they appear in the blood by 3 days, and they will peak by 2 to 3 weeks of starvation. So that's all about integration of metabolic pathways during fasting and starvation. Thank you.